Hello and welcome to the Parkinson's Foundation Wellness Wednesdays webinar. The Parkinson's Foundation is a dedicated nonprofit organization committed to enhancing the lives of those living with and impacted by Parkinson's disease. We focus on improving care and advancing research, ensuring that all our efforts are closely aligned with the needs and priorities of the Parkinson's community. While still in the developmental stage, genetic and cell-based therapies hold some promise for people with Parkinson's disease. Today's webinar will review the basics of gene and cell-based therapies. We will discuss what these therapies may or may not accomplish for the person with Parkinson's, as well as what to expect in terms of the treatment process. Please note that the nature of this topic is scientific, and we will be sharing more technical terms throughout the program. Join us this November for our Care Partner Summit webinar series designed specifically for those of you who are supporting someone with Parkinson's. Whether you're new to the role or have been caring for a while, these webinars will offer valuable strategies for managing Parkinson's, maintaining strong relationships, and navigating daily challenges with confidence. Our expert panelists will share insights and practical tips to support you every step of the way. We are hosting 3 webinars throughout the month of November. Sign up for all 3 or the one that speaks the most to you and your needs. You'll learn more details about these programs by visiting the website at parkinson.org/summit, or take out your smartphone and scan this QR code on the screen. We hope you will join us for these webinars designed specifically for those of you supporting someone with Parkinson's. To begin our formal presentation, I'd like to introduce our presenter for today's webinar. Dr. Joohi Jimenez-Shahed is an associate professor of neurology and the medical director at the Movement Disorders Neuromodulation and Brain Circuit Therapeutics. Dr. Jimenez-Shahed is co-chair of the executive committee of the Parkinson Study Group, where she previously served as chair of the mentorship committee and of the Functional Neurosurgical Working Group. She currently serves as co-chair of the Medical Advisory Board of the Tourette Association of America and on the board of the International Association of Parkinsonism and Related Disorders, and also on the Council of the Deep Brain Stimulation Society. She recently completed a term as chair of the Science Advisory Board of the Davis-Phinney Foundation for Parkinson's and previously chaired the Medical Advisory Board for the Houston Area Parkinson's Society. Dr. Jimenez-Shahed, thank you so much for sharing your time and your expertise with us today.
I had to unmute. Thank you so much, Krista, for that excellent and wonderful introduction. Let me go ahead and just put my slides up here. But while I'm doing that, I just wanted to thank the Parkinson's Foundation for having me, and I'm pleased to be sharing this presentation with you today about gene and cell-based therapies. So I'll just start out by giving like a little bit of my personal background. I started out in this field about 20 years ago. And when I started taking care of patients with Parkinson's disease, this was starting to be kind of a hot topic, and especially things like stem cells. And I remember that patients were often asking, what's the deal with stem cells? When are they going to become available? At the time, again, remembering this was about 20 years ago, I would say things like, 10 years from now, we'll have something. So 10 years came and went, and we still didn't have things to really talk about in terms of major advances. And then another 10 years went by, but here we are. As I was doing kind of the research for this, it was kind of really nice to be able to look over the degree of progress that we've had over the past 2 decades in terms of bringing some of these therapies to really good clinical trials. And that's the information that I hope to share with you today. So just briefly, some disclosures. I have done some work with Blue Rock Therapeutics. They have, through consulting and also by serving on their data safety monitoring board, also with Medtronic, MLX, Photopharmix, and a couple of other drug companies here who will not be represented through the course of this talk. So as Krista briefly mentioned, the objectives for today's presentation are as follows, and I'm really just going to structure the talk along these lines. So first, we'll talk just a little bit about some basics of gene and cell-based therapies. Then we'll move on to learning how some of these gene and cell-based therapies may or may not help patients with Parkinson's disease. And then lastly, we'll discuss the specifics of actual gene and cell-based treatments. So I'm not going to spend a whole lot of time on Parkinson's disease itself. I hope that that is a topic that you all are familiar with. If there are any questions about that, I'm happy to try to answer them in the context of how it relates to the cell and gene-based therapies. But we are going to just start right off the bat with some basics of gene and cell-based therapy. So first of all, what are these? We're using these terms, but it's probably good to just level set and make sure that everybody's on the same page. When we're talking about cell therapies, these are the ones that are on the left-hand side of this image here. These are where cells that maybe have a particular function or a particular characteristic are implanted into a patient with, let's say, Parkinson's disease or other disease or disorder, in order to kind of replace some other cells that aren't functioning properly, or to sort of have a particular type of function that may not exist in those patients. When we are talking about these cells, there's different kind of sources of the cells that we can think about. And the terms that are listed here are autologous and allogeneic. So autologous cells are those that are actually derived from the patient who is then going to go on to receive that treatment. So a person's own cells are maybe banked, then they're maybe sort of modified in some way or treated in some way prior to reimplantation back into that same patient. Allogeneic cells are those that come from a patient at some point. It could be a person with the disease. It could be a healthy person that was the original donor. But then these are similarly kind of modified and expanded. More cells are made, and then those cells are implanted into multiple patients. Okay, so that's kind of one of the main differences between autologous and allogeneic cell therapies. Now, on the right-hand side of the slide here is a picture sort of depicting gene therapies. So what are gene therapies? These are treatments that are geared towards inserting or creating a new gene with a new function in a person with a disorder such as Parkinson's disease. So there may be certain components of that person's genome that are not functioning properly, or maybe producing aspects that are relevant to why the disease is happening. And by inserting a new gene into a person with that kind of condition, we could either replace that function that is not going very well, or to, again, enhance certain functions that might not be working properly. Sometimes even repairing a mutation. These are different ways that gene therapy can be done. So if we kind of break this down a little bit further, first, in terms of cell therapies, there are different kinds of cell therapies that are already out there in the world. So one is hematopoietic stem cells. That means stem cells that are capable of making some sort of blood cell or blood product. One common example of that many people may have heard about is bone marrow transplant. So this is already a form of cell therapy that exists and can be delivered to patients. I'm not necessarily saying Parkinson's patients, I'm just saying in the space of health and disease. These are the types of cell therapies that exist. Other types of cell therapies are things like immune system cells. And an example of that some people may have heard about is the CAR T-cell therapies that are being used for cancer, and kind of taking advantage of some of these cell functions and the way that these cells kind of work and kind of do, what they're otherwise intended to do to actually fight the cancer. Another type of cell therapy that is already available and can be used in different ways is mesenchymal stem cells. And these are a particular type of stem cell that is attracted to inflammation that is occurring in the body. And there are different uses or proposed uses for that treatment. And then the last one is what we call pluripotent stem cells. And that is the type of stem cell that has the opportunity to be coaxed into becoming a number of different kinds of cells based on the way that it is treated, the nutrients that it's given. And this is probably the focus of where we will spend our time today talking about cell therapy in Parkinson's disease, because this has been the area that has seen sort of a lot of the most advances in recent years. And then, as I mentioned already, the sources of these different kinds of cells can be autologous or it can be allogeneic, as I already defined. So when we're talking specifically about pluripotent stem cells, another sort of way that you might see these referred to in the literature is as an iPSC. So PSC is for pluripotent stem cells, and then the I part of it is that it's an induced cell. And basically what this means is that there's an entire process that occurs in order to harvest the cells, to treat the cells, to get them to differentiate into the specific type of cell that we ultimately want to implant in a patient. So an example of this would be, let's say there's a patient who is able to have a part of their body biopsy, commonly something like a skin biopsy, maybe some blood cells could be taken that also have stem cell or that have cells within them that could be harvested in this way. So either the patient or some donor would kind of be the source of the cells to begin with. And then those cells are reprogrammed to kind of revert back to an older way of being called a stem cell, similar to an embryonic stem cell, but not exactly. But they're just brought back to a phase where they can then, like I said, be sort of coached and reprogrammed to become a particular type of cell. And that's what's on the third panel here. Once you take a kind of adult cell, make it into a stem cell, you can then cause it to differentiate into a particular type of cell, like let's say a liver cell, or maybe a nerve cell, maybe a heart muscle cell. And then those specific types of cells can be implanted into a patient to treat any specific disease for which those cells are relevant. So that's kind of the process of an induced pluripotent stem cell and making that cell into something that we want to implant into a person. So how is that relevant to Parkinson's disease? Well, again, you may have a donor or a patient from whom a biopsy is taken. The adult cells, sometimes things like skin fibroblasts, as I mentioned before, are reprogrammed into actually becoming stem cells. And that pluripotency then can be further differentiated. Those cells can be further differentiated into a functional cell of a specific type. And a logical thing in a patient with Parkinson's disease, which we know is symptoms of Parkinson's are often coming from loss of the cells in the brain that make dopamine. So a logical kind of thought would be to create dopamine producing cells eventually for transplantation into a patient. Now, sometimes it's just a process of harvesting the cells, making them into stem cells, differentiating the cells into dopamine cells, or you could modify them a little bit by gene editing and then create the cell and the cell characteristics that you need or want. So sometimes that process is also done. But then ultimately, when you have the cells and they're growing properly and they are kind of producing the things that we want them to produce, then they can be implanted into a patient. And some of the advantages of this kind of cell therapy are that it's a one-time treatment. You don't have to keep going back for multiple treatments in order to get the benefits. And there may be a number of different functions that can be reintroduced into the patient with Parkinson's disease in order to restore different aspects of brain functioning. And we'll talk about what some of those different ones are. So we talked mainly about dopaminergic neurons, but there's other ways that this can be done as well. So if we turn for a moment to gene therapy, that's a little bit different from cell therapy, because here we're not taking an entire cell and implanting the cell. Here we're really kind of trying to reintroduce a specific gene. So genes encode proteins, and there are sometimes genetically abnormal proteins that can be associated with disease. And like a way to think about this in Parkinson's, for example, is maybe the alpha-synuclein protein. That is a protein that is abnormally accumulating. This is just an example, but a gene therapy might be a way to transfer, or gene therapy is a way to transfer a new functional gene into a patient. And that's different from gene editing, where you're just trying to correct the dysfunctional gene that already exists. So I just want to make that distinction between gene editing and gene therapy. What we're going to focus on here is gene therapy. So when you're doing gene therapy, this requires certain kind of equipment, if you will. It's not just that you have a bunch of genes in a test tube or in a syringe and you inject them. This actually has to be delivered into very precise areas of the body, and in the case of Parkinson's disease, into certain parts of the brain. And so the most common way of trying to deliver a gene therapy is through taking advantage of other molecules or other organisms called just viral vectors. So some of you may be familiar with the adenovirus, which is a flu-causing virus, but there's several different strains of adenovirus that actually don't cause sickness necessarily, but they have all the machinery of a virus that can get into the body, can get into the cells, and can deliver the products that they have. Now, most viruses carry some type of genetic code. And so we can insert the gene and the gene code that we're primarily interested in into this viral vector, so kind of the shell of the virus. And that shell can be further modified so that it has specific ways of interacting with the body or with the brain in particular, when we're talking about Parkinson's disease. And that is how we can get these virus particles containing the new gene into very specific parts of the brain, for example, and to deliver the product, which is also often referred to as a transgene. So the transgene is the functional version of a defective gene that is carried within the viral vector and delivered to the brain of a patient with Parkinson's, for example. All right, and so this is just another kind of picture of how that process occurs. On the left-hand side here, if you look at this picture, there's a virus vector. It has the gene inside it. That vector sort of stays in there while the virus itself fuses with the cell in the brain. It then makes its way into the nucleus, and it can deposit the new gene into the nucleus of the patient. And that new gene then has the opportunity to kind of integrate and to do what we want it to do. And we'll explain a little bit through some specific examples in the coming slides. All right, so how can gene and cell-based therapies help? Well, when we think a little bit about just sort of how we generally manage Parkinson's disease, many of you are familiar with all the different medications that are listed on the left-hand side of this slide. So the primary way that we have of treating symptoms is to give medications. And as you are probably well aware, this often has to be changed on a regular basis so that people are taking more and more medications over time. And along with taking more medications, there can be risk of side effects. There can be risk of developing the motor complications, especially with levodopa, where medications are kicking in, they're wearing off, maybe they're not controlling symptoms adequately throughout the course of a day. And these are just some of the known challenges of how we manage Parkinson's disease. And when we get to some of these more advanced stages where a lot of these fluctuations, dyskinesias, et cetera, are occurring, then there are certain advanced therapies that can be considered, things like deep brain stimulation, perhaps focused ultrasound for some qualified individuals, a levodopa pump, of which there are now a couple available. So these are some of the strategies that we have to kind of manage it, but that's really kind of the key. This takes a lot of management, a lot of long-term follow-up, a lot of adjusting, a lot of knowledge of how these different medications kind of work together, and when the timing is right to move on to a more advanced therapy. So this is kind of the landscape that we kind of are currently in. But with these new cell or gene-based therapies, what we're really kind of talking about here is being able to intervene at a single point in time to deliver either a cell therapy or a gene therapy. And through those particular therapies, be able to start addressing some of the problems that the medications aren't addressing, maybe preventing some of those complications. And if they're doing a pretty good job of helping to treat the Parkinson's symptoms, maybe allowing patients to reduce the amount of medications that they need to take for their Parkinson's disease. And so this is kind of the promise of cell or gene therapies, or the kind of reason why these are so attractive as potential possibilities, because it can really, as we'll talk about, change, if they're successful, change the way that we approach the management of Parkinson's disease with our otherwise medication and surgical options. So here's a brief history, very brief, of cell and gene-based therapies in Parkinson's disease. Some of you may be familiar with some of these elements, but I'll just kind of try to set the stage a little bit for demonstrating the progress that we have had in these areas in the present day. So back in the 1980s, some of you may have heard about or maybe have read about the original cell transplants that were done in Parkinson's disease. These actually were adrenal cells. So the adrenal gland is a gland in the body that contains neurons that create epinephrine and norepinephrine, and probably to a certain degree, there is also some dopamine production in there. And it was thought that by transplanting these cells that were capable of producing dopamine into the brain, that we could take a person with Parkinson's whose brain isn't making enough dopamine to maybe enhance that dopamine production. And so there was several different attempts at trying to do this, some more successful than others, maybe varying degrees of success, a lot of heterogeneity, but really not super kind of effective across the board in terms of helping to treat Parkinson's symptoms. In the early 2000s, we started seeing the transplantation of embryonic dopaminergic neurons. So if you took a embryo and were able to differentiate some dopamine neurons out of that, and then later in that same decade, there was also the fetal nigral cell transplantation. So this is the place in the brain where the dopamine cells originate. There were a number of these trials that also went on, again, with varying degrees of success in terms of being able to successfully get into the brain, to integrate into the brain, and then to even really do the things that we wanted these cells to be able to do in order to help control Parkinson's symptoms. So then in the later 2000s, we started seeing things like GDNF infusions. GDNF is a neurotrophic factor. It is important for the health and survival of dopaminergic neurons, amongst other neurons in the brain. This was originally attempted as an infusion, but there were other delivery methods that were also explored over the years. Again, a lot of different challenges associated with this type of treatment, but the thought was that if we could deliver these neurotrophic factors, that could help prevent the cell death, maybe slow down the progression of Parkinson's disease. And unfortunately, again, a lot of challenges that we really had some difficulty kind of getting over. And then in the 2010s, we started seeing gene delivery of those same neurotrophic factors. In this case, the nurturin. Nurturin is another neurotrophic factor that was delivered via this type of virus vector that I explained to you earlier. And all of these things were kind of met with, like I said, a number of different challenges, sometimes some disappointing results. And I think the question really became trying to get a better understanding of why there were so many difficulties using these types of therapies, because the science seems really good. It seems solid. It seems like these things should be able to work if we can get them delivered the right way, if we can get them into the brains of patients who need them and get them to do, kind of what we want them to do. And I think alongside all of these kind of attempts have been some advancements in the surgical techniques that we have to actually deliver these therapies. And I think this is an important point to kind of make. Neurosurgery these days is very different from the neurosurgery that we did 10s, 20s, 30s years ago, in terms of the equipment that we have, in terms of the guidance that we can use to really pinpoint the correct areas of the brain to deliver these therapies, even just the cannulas and the injection techniques that we have for the cells and the gene-based therapies, how to do that efficiently and to do that safely, and to, again, just really kind of streamline the surgical process to make sure that that is being done in the best way possible. So we've had a lot of experience doing these types of cell and gene therapies in the past, and I think we're really just at a point now where we have a much better understanding of how all these things should work, what are some of the things we can do to optimize the outcomes, and then to really kind of deliver these therapies and start really studying how they can be effective in patients with Parkinson's disease. So when we think again for a minute about kind of the opportunities and risks, with cell therapy, I think, as I mentioned before, one of the big, sort of dreams here is that we can replace the dopaminergic neurons that are degenerating in Parkinson's disease, that we can deliver them to the site where they are needed, in the parts of the brain that really rely on the function of these neurons. And while we do that, to maybe take the place of some of the medications and reduce the medication requirements, and as a consequence, reduce some of those medication side effects. And then to really kind of get those dopamine cells to function in a way that is more normal. So the way that we give levodopa and dopamine therapy in Parkinson's disease is not the way that the body is used to receiving it or used to needing to try to react to it. We give these very high doses intermittently multiple times throughout the day. And that's just not how the dopamine system was designed to work. And so if we can get these cells to do it in a way that's just much more natural, then perhaps we mitigate some of the issues with long-term levodopa therapy, like the dyskinesias and the fluctuations. And through doing that, perhaps all of these different steps allow us to kind of modify the disease course, or at minimum, modify the way that we need to kind of treat the condition and hopefully get better function and better outcomes for our patients. Now, there are a number of different risks that can come with these types of therapies. There have been concerns, sometimes even reports of some of these things in the past where the cells are less regulated. Maybe there's tumor formation or risk of tumor formation. Maybe the cells are moving into different areas and kind of now exerting their effects in parts of the brain where we don't want them to do that. Perhaps there's an immune response to those cells. There's obviously the risk of the surgery itself for the implantation. And there really has to be a very solid process for handling those cells prior to them being transplanted so that we're not doing things like transmitting risk of infection. Other challenges that I think have come clear through all these sort of decades of experience that we have had in doing these therapies are understanding who the right patients are for the treatment. And some of our data definitely suggests that people who are younger, maybe people with a better levodopa response, or patients with certain kind of degrees of pathologic change in the brain might be better suited or might be better positioned to have a better response to some of these therapies. Another kind of challenge is making sure that the tissue that we're implanting, the cells that were implanted, are able to survive, and not only to survive, but to do the things that we want them to do, as I've mentioned before, in a kind of well-regulated way. And another challenge that was kind of not expected, but a sort of long-term effect that was seen in some of the early studies was this idea of graft-induced dyskinesia, where patients were experiencing dyskinesia even in the absence of taking medication. And that was a direct result of the cells that were implanted into the brains of some of these patients. And so trying to understand more about why that happened and what we can do to avoid that, these are all some of the challenges that people and scientists have tried to understand over the years and have incorporated into the current designs. Now, another just word about some of the cells that we have tried to use in the past. They've tried to use fetal tissue, mesencephalic tissue, the adrenal tissue that I talked to you about before. There were even some reports of carotid body tissue transplantation. Another study that I had an opportunity to participate in previously was the retinal pigmentary epithelial cells, which can also manufacture some dopamine. There was a attempt to use that to transplant those into the brains of Parkinson's patients. And then there were also some porcine sources of cells as well that were also studied. Again, with varying degrees of success and certainly a number of different challenges in terms of how these tissues are supplied, how they're prepared, how they are kind of delivered and delivered at scale for treating large numbers of patients with Parkinson's disease. So in order to kind of really overcome some of those challenges, there has been more and more attention turned towards the use of different types of stem cells. And what I talked to you about earlier was the induced pluripotent stem cell. Some of you may be familiar with the concept of embryonic stem cells as well. And then there's another form of stem cell called a parthenogenetic stem cell, which is made from a egg cell or derived from an egg cell that is not fertilized yet, so not an embryo, but prior to that stage. So these are different sources of stem cells that can be used. And the advantages really of stem cells are the availability, because they can multiply and kind of make more cells of the same type very readily. These can be much more widely available at the numbers that are needed in order to treat patients with Parkinson's disease. We can also standardize their manufacture because they really are sort of cultured and manufactured. They can be frozen and preserved and then be made available when they are needed. There's much more that we can say and do about maintaining the purity of these cells. And the source of the cells is really kind of a single source that can be sort of kept kind of pure from that standpoint over time. And then because of all of these things, we have a much better opportunity to really investigate the best ways to do the surgery, the best way or the right kind of dosing for these cell therapies, and even the distribution of the cells. So all of these things can be done a lot more homogeneously in a rigorous way to be able to study the effects and not have to worry about any of these things kind of complicating our interpretation of the results. So we've talked about the opportunity of gene therapy to increase dopamine production. We have talked, or there's other ways that gene therapy can be used. They could be used to deliver genes that support the survival of dopamine neurons by delivering these neurotrophic factors that I mentioned before. There's also a couple of other ways that gene therapy can be used. One is to try to reset the abnormal brain circuitry, and there's a specific type of gene therapy called GAD gene therapy that has been investigated in this way. And then there is also the opportunity through gene therapy of being able to counteract genetic mutations that contribute to Parkinson's risk in certain forms of Parkinson's disease like GBA or LRRK2. So all of these are different ways that gene therapy can actually be used in the treatment of Parkinson's disease. There are different challenges with gene therapy that are different from the challenges that I mentioned with cell therapy. So gene therapy, because it has to be delivered through this vector, we have to be able to get that vector across the blood-brain barrier, and either that's done surgically or we have to find ways of making that vector and that capsid, the protein that kind of is around the genetic material that we're trying to get delivered. We have to make that a little bit more specific and help us really kind of get the content of that vector across the blood-brain barrier and into the brain where we need it. There's also been some work investigating how this could be done through an infusion as opposed to a surgical therapy, but that also remains a challenge. We also want to be sure that we're able to deliver this gene therapy to the intended target, but without affecting other brain areas or cells. And some of the ways that this challenge has been tried to be kind of navigated is through specific viral vectors. There's different types of these, and so some may work better than others. Again, there's also different things that can be done to that encapsulation design to help attract it to specific brain areas. Another challenge is making sure that there's long-term and stable gene expression. Once you get the gene in, it has to be able to kind of do its thing and produce its products without an immune response or without toxicity. And so then there's been some thinking about the type and the duration of immunosuppression that might be needed in order to minimize that. And again, specific virus types may be more amenable to this than others. And then similarly to one of the challenges with cell-based therapy is really the optimal patient selection criteria, because Parkinson's, as we know, is quite heterogeneous. And so which are the patients that are likely to benefit from a specific type of gene therapy? So this also requires some thought. So moving on from that, then we'll spend the next chunk of time here talking about the cell and gene-based treatments that are currently under investigation. So I really just kind of want to walk you through this. It may seem like a long list, But what I actually think about that is that that's a good thing, because that means that there's a lot of really good science and a lot of good research that is going on. And I think what you'll see is that we're really at a point where we can find out some really important information that may really allow us to advance our treatment of Parkinson's disease. So let's try to go through these one by one. The first is just a summary slide that shows you the different types of products that are currently under investigation. You'll see 3 gene therapies on the left-hand side of the slide and about 6 cell therapies that are on the right-hand side. So that is the information that we will work through now one by one. The first one that I want to mention to you is a product called bemdaneprocel. And this is a cell product that is developed by a company called Blue Rock Therapeutics. And what this entails is a one-time surgical implantation of a dopaminergic neuronal progenitor cell that is targeted into a specific part of the brain called the putamen. This is one of the important areas that requires that dopamine cell input. And these cells are derived from a human embryonic stem cell line, and they have been differentiated into these very specific progenitor cells that are capable of making dopamine. And the idea with this particular cell treatment is to replace dopaminergic signaling in patients with advanced Parkinson's disease. Now, the good news here with the bemdaneprocel is that they have now completed a phase 1 clinical trial. This included 12 patients who were followed up for a year. These were patients who were aged 50 to 78. They had Parkinson's motor complications that were inadequately treated by standard medication management. And they investigated a low dose and a high dose group. There was not a sham procedure in this early phase study. This study, because it is a cell-based study, did require the use of immunosuppression for the first 12 months. And then they looked at the primary outcome which was safety actually, at that 12 months to see whether patients were able to tolerate this procedure or whether there were any other kind of unanticipated risks. So what they found in these 12 patients is that bemdaneprocel was generally safe and well tolerated, and there weren't any significant or serious adverse events that occurred as a consequence of the procedure or as a consequence of the cells. And they saw that they had slightly kind of new equipment that they used in order to deliver the cells, and they were able to show that this was feasible, that it was able to be done, that it was able to be done safely. And again, that patients were able to tolerate not only the procedure, but then also to tolerate that 1-year immunosuppression regimen. I think from a neurology standpoint, it was also good to be able to see that the immunosuppression regimen was relatively safe. There's protocols to kind of handle that appropriately, and the neurologists were able to do that, and the patients were safe with that as long as the protocols were being followed. What they were also able to show in this early phase 1 study is that the cells from the bemdaneprocel were surviving, and that they were actually really engrafting and incorporating into the brains of patients where they injected it. So it was kind of staying where it needed to stay, it was doing what it needed to do, and it was integrating with the normal brain tissue. And then on a number of different sort of exploratory endpoints, they did see some changes in motor symptoms of Parkinson's disease that really seemed to happen in a dose-dependent way. So the patients that got the higher dose potentially seemed to be doing a little bit better than patients who got the lower dose. And of course, this is a phase 1 study. It was only 12 patients, so we have to really interpret this with caution. So we can't get overexcited about what those benefits are going to be, but we can definitely see that there is need to continue studying this therapy. It can be done safely, and now we have the opportunity to move on into more advanced phase studies to really see what the clinical benefits can be for patients with Parkinson's disease. So that was the first cell therapy. Another cell therapy study that is ongoing is called the STEM-PD study. And here they created a human embryonic stem cell master bank, or they got cells from a human embryonic stem cell master bank. They tested those extensively and then differentiated those embryonic stem cells into dopamine progenitor cells. And then they really took a moment to test those cells extensively in animal models and on the bench. and eventually were able to get enough sort of regulatory approval for the implantation of these cells into humans. And the group of patients that they chose to treat here were patients with moderately advanced Parkinson's disease. You see that I put that in quotes because these are not absolute terms. But the patients who received this therapy also required immunosuppression for the first 12 months after their graft. All of these procedures, or all the patients, were in Sweden and in the United Kingdom, but all the procedures were done in Sweden. And they were able to get the first 4 patients implanted, and we did a safety review and got approval to be able to continue into the higher dose cohort. And that approval was obtained in May, and they're planning to implant another 4 patients and to measure their safety and some other Parkinson's disease related measures, just to sort of see exactly how patients do. So I think another promising study, still in very early phases of cell therapy, dopaminergic cell therapy in Parkinson's disease. Now, the next one is a study from the International Stem Cell Corporation, where here they used a different type of stem cell. This was the parthenogenetic type of stem cell that I mentioned in the earlier slide. So these cells were kind of created and have the ability to differentiate into dopaminergic neurons and to express more of the neurotrophic factors. So they're not necessarily going to deliver more dopamine, although how kind of ultimately the effects of these treatments play out is still to be seen. But what they started out with was a single center dose escalating open label study in Australia. Again, 12 patients, so very small numbers here just to demonstrate the safety. These were patients with kind of a broader sort of kind of disease range. They could have, as long as they had Parkinson's for less than 13 years, they had to have some evidence of motor fluctuations with at least 2 hours of off time, and some other specifics here in terms of their staging and their motor symptom severity. And they injected these cells into the striatum and into the substantia nigra. So you're seeing now slightly different locations of the injections. And interestingly, this is a cell therapy that they plan to study in other disease states, including traumatic brain injury and ischemic stroke. So it's not a treatment necessarily specific for Parkinson's disease, but can be used in other disorders that also have evidence of cell damage. And again, this is very early. We don't have any data yet on how these patients have done, but this is an ongoing trial. Another study that some of you may have come across is from a group called Hope Biosciences Foundation. And here, yet another type of cell therapy. This is a mesenchymal stem cell that is being studied for multiple different kinds of indications, so not just Parkinson's disease. But mesenchymal stem cells are a little bit different. They're not differentiated. We're not trying to differentiate these into other cells that do other things. But these are the type of cells that are attracted to inflammatory hotspots and may help by secreting more of these trophic factors, other sort of molecules that can kind of help calm down the inflammation. And there may be a number of different reasons why that might help in Parkinson's, but there also may be a number of different disorders in which this type of treatment could be potentially helpful. And so this group conducted a randomized double-blind single-center phase 2 study to look at the safety and efficacy of this type of treatment. Now, a difference with mesenchymal stem cells is that it has to be administered more than once. So this is not the single cell or single treatment cell therapy that I was talking about earlier. This was all done at a center in Texas. The patients got 6 IV administrations, so that's another difference. They don't have to be injected or implanted into the brain. And what they were primarily looking at was the activities of daily or motor activities of daily living on the Parkinson's disease rating scale. And unfortunately, this did not meet that primary endpoint, but they did see some other indicators on non-motor symptoms of Parkinson's disease that seemed to favor the group that actually got the active treatment. So there might be more information here to be had. I know that there are other companies out there that are looking at mesenchymal stem cells or other investigators that are looking at it. And so we may still find more information about this particular treatment in the future. The next one is by a company called Sumitomo. This is a Japanese company. They are doing a phase 1/2, so again, early phase study that is actively recruiting. And here they're looking at allogenic human-induced pluripotent stem cells that are dopaminergic progenitors, so they're capable of producing dopamine, and then surgically implanted into the striatum of patients with Parkinson's disease. So again, here they're looking at a slightly different number of patients, 7 patients with Parkinson's for 5 years. Again, very specific staging criteria, and they're patients who are not receiving an adequate response to their medication management. Also, this treatment will require immunosuppression, also very early days, and we will look forward to seeing the results of this study as well. Now, Aspen Biosciences is another company that has yet again a different type or a different approach. And here, this stem cell therapy is actually from patients' own cells. So here, patients are kind of start by getting sort of a skin biopsy, and these cells are then reimplanted into that same patient. So there's no requirement for immunosuppression, but they do take the skin cells from the patient, reprogram them into an induced pluripotent stem cell, and then differentiate them into the dopaminergic neuron precursors. And then those cells will go on to produce dopamine, hopefully in the brains of patients in who they are implanted. And so this company not only is doing this autologous approach, but also has a kind of platform that allows them to really kind of test the integrity of those cells and the quality of those cells, and really kind of keep track of all of that in a very precise manner over kind of the different numbers of patients that are being enrolled in the study. So these this company has started their early phase study. Their product is called ANPD-001. Again, they're looking at safety and tolerability of 2 different doses of this product, and they're going to follow patients for 5 years, hopefully even on to 10 years if they're able to do so by continued phone calls. And again, they are just in the recruitment phases. They started in April of this year, and we'll be looking to hear more from them in terms of the outcomes. So those were the cell-based therapies. Now we've got the 3 gene therapies that were also on that kind of opening slide there of the ongoing trials. So the first one is AB1005. This is being developed by a company called AskBio. And here they are trying to deliver the gene for GDNF. And some of you may be familiar with kind of the GDNF story over the years. I kind of alluded to it in that history slide. But there's been a long history of trying to get this particular neurotrophic factor into the brain of patients with Parkinson's disease to see if we can, through delivery of this neurotrophic factor, help prevent the cell death, potentially slowing down the progression of Parkinson's disease. So these guys, again, are using an adenoviral vector to deliver the GDNF gene. And by doing that, we know, like at least in the laboratory, that increasing GDNF expression leads to functional recovery of those dopamine neurons and even regeneration of those connections. So it sounds like a really, kind of nice idea. I think there's a lot of science supporting this approach. The challenge, again, over the years has been really being able to deliver it in the best way possible to the right patients, and being able to see then the results. So this company has also completed its phase 1 study of this therapy. Again, they were looking at patients 35 to 75 years old, relatively earlier in their course of Parkinson's disease. They could be stage 1 to 3 off medication. But they did divide these patients into 2 cohorts. So one that had milder disease and one that had more moderate disease. And you can see the different descriptors there of the specific cohorts. But we do have their early stage results, which showed again that this type of infusion or infusion into the brain was well tolerated. There were no significant adverse effects. And most of the patients in the mild cohort kind of stayed stable, whereas patients in the moderate cohort actually showed improvement or stability on several motor scales and Parkinson's disease motor diaries and other indicators of Parkinson's disease control. So based on these results, that it was safe and that maybe there were some clinical indicators, this will move on to the next phase of investigation as well. Another company that has a gene therapy that has kind of completed some of the early phase studies is the GAD or GAD. Glutamic acid decarboxylase is an enzyme that increases production of a different brain chemical called a different brain neurotransmitter called GABA. And GABA is inhibitory. So if you kind of have GABA in the midst of a bunch of neurons, it will actually suppress the activity of those neurons. And so what they have, this is again, has been a line of investigation that has been ongoing for a number of years with studies dating back even to 2003. So there was a phase 1 study in 2003 and a phase 2 study in 2008. So this is really just kind of iterating and trying to figure out the best way to do this type of gene therapy. But the idea is that if you can get into, in this case, the subthalamic nucleus of the patient with Parkinson's disease, and you deliver this GAD gene therapy, you can sort of suppress the output of the subthalamic nucleus. And this is an important node in all of the motor circuitry of the brain that controls movement that is also targeted, actually, interestingly, by deep brain stimulation. And so similar kind of concept in terms of the circuit approach to Parkinson's disease, but here doing it in a gene therapy way as opposed to, let's say, a stimulation way. And so what they did in 2022, they started their study of patients with Parkinson's disease. They had a low dose, a high dose, and a sham infusion. They ended up being able to enroll 14 patients, and they found that the treatment was safe and well tolerated. And that, again, there were some positive indicators of motor improvement in the patients who got the active treatment. And there was also some improvement in quality of life. So again, I think we're going to see this particular therapy move on to more advanced stages of investigation. The last gene therapy that I'll mention here is the Prevail therapy, or a company is called Prevail, and the therapy is called a gene therapy for GBA1. And this is building on the fact that in patients with GBA Parkinson's disease, the glucose cerebrosidase activity is reduced. And here we can deliver a GBA1 gene that will then produce normal amounts of this enzyme and hopefully can help reverse symptoms in that way. And so this is done slightly differently. We're not implanting it directly into the brain. It's actually being infused into something called the cisterna magna, which is a fluid-filled region just under the cerebellum and behind the brainstem. It uses a slightly different virus vector. And the group of patients that are being looked at here are those with moderate to severe Parkinson's disease who have at least one GBA mutation. Again, this treatment will require immunosuppression, and recruitment is ongoing. So we'll see whether this particular approach is safe and whether it's effective, and more data to kind of come from that. So I know that was kind of a long list of different studies, but I hope what I've shown you is that there's a lot of really good stuff going on now and a lot of really well-designed trials. And we're really at that point where I think we can then get some really good information about how these cell and gene therapies can work in Parkinson's disease. I think that these therapies offer different and unique ways of addressing the underlying abnormalities in Parkinson's. Some of them are going to be more broadly applicable to large groups of Parkinson's patients. Others are maybe going to be more targeted towards specific groups of patients with certain characteristics. as you saw here, there was a wide range of study design, a wide range of inclusion and exclusion criteria. So we're going to have to work to make sense of all of this stuff once we start getting in more data. And all of these are, I just want to emphasize that they remain under investigation. So it's still going to be some time before they are available. But so far what we know is that they have a reasonable safety profile in small numbers of patients, and they're worth studying further to really kind of understand how they can help. And we really do need that long-term data in order for us to make more definitive conclusions about their applicability and about their benefits. The last thing that I have on this slide here is just a reminder about the process of developing new therapies. It's a lengthy process, so patience is certainly required, but I think being able to do these studies in a very well-designed way, in a carefully controlled way, really allows us to get some useful answers that will help push the field forward in terms of bringing new therapies to patients with Parkinson's. So I will stop there, and I would be very happy to take any questions in the few minutes that we have remaining.
Thank you, Joohi Jimenez-Shahed. Really appreciate all the time that you've put into creating this slideshow and recognizing that this is a subject that is still under development, and we're still learning so much, and we've already learned so much, and more to come. So thank you for highlighting what's possible and what's happening in the field of gene and cell-based therapies for people with Parkinson's. We have one question from a participant, Bob, who asked, what is the state of the current study using autologous stem cell neurons injected into the brain? Can you speak to that?
So I believe that one was the Aspen study. That was the one autologous dopaminergic cell therapy that I mentioned there. So I did mention that in one of the slides. So if these are made available later, then maybe that can be referenced. But that study is ongoing, and I think that it is a different approach. So what we really have to kind of understand about this approach is, because it's individualized, right? A patient is getting their own cells back. I think the question about how scalable that is going to be is going to be one of the big questions. And so patients are actively being enrolled, and we'll see what the data show us.
Waiting for that data. And for data, we need participants. So just joining opinion. I have a question from Alan, and they ask, can these therapies be given to a patient who has had deep brain stimulation?
Yeah, I think that's a really good question. Right now, because all of these therapies are investigational, the requirement will be that patients have not had any kind of brain intervention before. So people who already have deep brain stimulators will not be eligible to participate in the studies. I think maybe part of this question is wrapped up into the idea of DBS as a therapy that's kind of available now. And if a person is maybe needing that type of treatment at the moment, we've kind of always talked about how DBS is reversible. You can stop it, you can take it out, and potentially when these types of therapies come out, patients could still be eligible for them. And I think, that is still a kind of a question that we need to answer. We believe probably that will be the case, but I think we just need more time to understand not only the safety profile here, but really what the outcomes are going to be, how that measures up to DBS in some ways. And is it really better? We don't even know answers like that just yet. So I think more to come on that, but still potentially a possibility.
Thank you for your response. And we have Jen who says, thank you for the presentation. It gives me hope for the future. And how can the Parkinson's community help support continued inquiry into gene and cell-based therapies? How can we support this research?
I think that's an excellent question. And, these types of therapies, I think, as I've talked about, have had their challenges over the years. But none of it would be possible without patients who are willing to participate in the trials. And I think, again, dating back to when I first started my career and thinking about some of the therapies that people were signing up for, I think a lot has changed. And we really kind of see this rigor to the way that the treatments are kind of reliably, kind of created and produced and manufactured. And we're also seeing rigor in the way that the trials are being done. So I actually now feel very confident in recommending that if there are qualified patients, that they could and should potentially consider enrolling if they are comfortable with the risks and benefits. And I think that's the number one thing is volunteering. If you are interested in that, advocating in other ways for the advancement of these therapies is also important. important.
And we have time for just one more question. Is gene therapy or stem cell therapy better than deep brain stimulation?
So I think that, again, probably dovetails on the earlier question a little bit as well. I think we don't really know the answer just yet. Some of the early studies that were done with these therapies, they looked kind of on a short time course, maybe 6 months, maybe 1 year. And probably what we know with some of these therapies is that it may actually take longer to get the results. So we have to look for longer, we have to follow patients for longer, and then we have to really kind of quantify what the degree of benefit is. And it may be that the benefits are a little bit different than what we would typically see with deep brain stimulation. It may be that we see more, it may be that we see less. I think time will tell us what we want to know there.
Great. Thank you so much for sharing your time and for all the efforts that you've put into researching genes and cell-based therapies for people with Parkinson's, Dr. Jimenez-Shahed. So thank you so much.
Thank you for having me. It's been my pleasure.
Today's program is sponsored by Blue Rock Therapeutics. Thank you, Blue Rock Therapeutics, for your support of the Parkinson's Foundation's mission. The Parkinson's Foundation funds basic science that leads to a greater understanding of Parkinson's disease. Our PD generation study provides genetic testing for PD-related genes and genetic counseling to people with Parkinson's, and this is at no cost for participants. Through PD generation, we're learning more about the genes related to Parkinson's. By including people from different backgrounds, researchers can better learn about the genetics behind Parkinson's disease, and understanding these genes and the differences between them can give us clues about why one person's experience with PD differs from another's. It can also lead to improved treatments and care for you and future generations. Participating in PD generation is easy. If you're eligible, you'll receive information you need to make an informed decision about whether or not to enroll. PD gene does require a blood draw. You can choose to participate in person at a participating medical center or from home through a telemedicine appointment and an at-home blood collection kit. Your results are shared with you during a follow-up visit with a genetic counselor. That's one of the key things that differs from our PD generation study and others is you get that counseling. It's very important in understanding the makeup of your genes. You should also know that there's no medications or long-term commitments, and your information is kept safe and secure through every step of the research process. So if you're interested in supporting the development and progression of furthering understanding the role of genes in the manifestation of Parkinson's disease, you can visit parkinson.org/pdgeneration or call our helpline at 1-800-4PD-INFO. And again, thank you, Dr. Jimenez-Shahed, for offering your knowledge and time for today's Wellness Wednesday. And a big thank you to each of you who joined us today. If you had a question that did not get answered, please don't hesitate to reach out to us. We have a comprehensive website, many more resources that addresses everything related to PD at parkinson.org. And again, that helpline is on the screen there for you to call. A wonderful thank you to today's program sponsor, Blue Rock Therapeutics. We appreciate the way that you support the mission of the Parkinson's Foundation. Thank you all for joining us. Until soon, be well, and may you continue to support yourselves in this journey through Parkinson's. Bye-bye for now.